Good morning and welcome to the second Biodiversity and Conservation Workshop. My name is Kathy Jacobs. I'm the director of the Center for Climate Adaptation, Science and Solutions at the University of Arizona. And I'm welcoming you today on behalf of the Arizona Institutes for Resilience at the University of Arizona. We want to point out to you that if you have questions or for the panelists during this event, please use the chat box and the answers will come during the discussion period. We are recording this event, so we want to make sure that you're aware of that. And we also have a poll for you that we will be opening now. We're hoping you'll all answer that while we're talking right now. We're just trying to find out a little bit about who the audience is before we start moving forward with our event. We'd like to acknowledge the fact that the University of Arizona occupies the original homelands of the Tohono O'odham and Pasquayaqui nations, indigenous peoples who have stewarded this land since time immemorial. The Arizona Institutes for Resilience has a vision to address the crises of climate and global environmental change head on, and to make the University of Arizona the go-to place for students and faculty committed to managing climate and environmental risks and building equitable and effective solutions to these challenges. Please feel free to go to our website, environment.arizona.edu. As far as today's agenda goes, we will start with speaker introductions. Paolo Quadri will be doing that. We do have 4 total speakers in 2 different sessions this morning with a short question and answer period in between. We'll then go into breakout sessions, and they are about 25 minutes long, small sessions, and then there'll be report outs from those breakout sessions. So be sure you're prepared to engage in those. There'll then be key takeaways, wrap up, and next steps at the end. The overall discussion question for the breakout groups today will be which processes, including colonization, extinction, speciation, etc., drive the biodiversity of different sky island systems? And how does this vary by taxa and by region? And how can conservation goals take these processes into account? So 3 pretty big questions, actually. So we'll then go to the poll so we can get the outcomes of that if somebody's able to do that for us. It looks like we have 40% early career scientist or professional, and then students and career scientists about 25% each. So this is actually quite a different makeup from our first event. And then in the second question about best describing professional background, the vast majority is scientific academic researcher, which is actually quite similar to the initial event. So we'll close that, and I will yield this back to Paolo.
Thank you, Kathy. Well, welcome everyone. Thanks for joining us today again. And so yes, please do note that the structure of the session has changed a little bit, so that we have these 2 sessions of questions and answers. So please take advantage of them as soon as we jump into them, so that we have more rich discussion. I'm super excited because, well, this is the topic that we'll cover today. This is admittedly the one that that I have almost no expertise. But as a conservation practitioner and a scientist, is extremely interesting. And so from Sky Island Alliance perspective, for example, one of the things that I'm excited to learn more about are things related to the questions of the impact of habitat fragmentation. Now, specifically, for example, dealing with the border wall problems that we're dealing in the area, but also urban sprawling, that we love in northern Mexico and we love in the United States, roads, and of course, climate change. And then another set of questions would be about restoration, like how much are we as conservation practitioners allowed to move and shift things around to help species cope with climate change in terms of like moving things from latitude and altitude and topography and helping them adapt. So with that, I want to introduce our two first speakers. First, we'll have Dr. Ela Vázquez. Ela is a molecular ecologist at the Instituto of Ecología of UNAM. She's been working on population and landscape genetics and genomics, phylogeography, hybridization, invasive species, and conservation of endangered species and natural resources. Her research mainly focuses on vertebrates in ecosystems ranging from tropical forests to deserts and from sky islands to the oceans, to actual real islands. And then we'll have Dr. Jessica Perez Alquicira. Jessica is a full-time professor at the Universidad de Guadalajara and cátedras through the program Cátedras CONACYT since 2016. Her main field of research is in population genetics and phylogeography, focusing mainly on plants, including agave, physalis, uadua, and brahea genera. So with that, Ela, welcome. You're the first one opening today. Thanks so much for joining us today.
Thank you, Paulo. It's ella. Vázquez.
Sorry.
Don't worry. Don't worry.
I have
just always
switch in brain.
I know. Okay, I'm gonna share my screen. Please let me know that you see it correctly. And I changed a little bit of my the presentation from the original title that you see there, in the aim of presenting to you two lines of research that we think they should be incorporated into landscape, into sky islands studies, because they allow to have different scales of time and space. And I will elaborate a little bit about that along the way. We've known for a long time, for example, by Alexander von Humboldt, that visited Mexico in 1803 and 1804, that mountains harbor an astonishing amount of the world's biodiversity in fauna and flora. And this is in part due to processes of speciation in allotry or paraptry that render this high species richness and high endemicity in these high mountains. And based on that, we should always realize or see the importance of integrating historical and contemporary factors when we're studying or evaluating these sky islands or these mountainous systems, in order to understand patterns of diversity among mountains and between highlands and the adjacent lowlands in the same mountain. And why this? It is because mountains are such complex ecological systems conformed by a myriad of climatic, environmental, biotic, and abiotic factors that all of them act in different spatial and time scales, from very historical and global, like latitude geology, to very local and contemporary. And we do have to integrate all this information in the research or the studies we do. The two research areas that I wanted to present today that deal with these different scales, and both of them rely on genetic information. That is key to this. One is phylogeography, that tries to decipher the historical patterns of diversification and differentiation among species along a long historical timeline, like here, and associates the biogeographical, geological, etc. processes that determine those diversification in species and populations. The other, in another more contemporary and local scale, is the landscape genetics, which the objective of this research area is to decipher how the heterogeneity of an environment, of a habitat, where a species lives, determines their connectivity, their dispersal patterns, their gene flow, which ultimately render how these populations within a species are differentiated along the habitat, along the ecosystem. With these two research lines, remembering sky islands, Paulo and the other presenters last Friday, they talked about how there's more, we usually think about sky islands in a temperate area, temperate region, but no, we also have tropical islands in, for example, Mexico, the Trans-Mexican volcanic belt that we both will be hearing about today, but even going south to southern Mexico and Central America, here we have like the Chiapas Massif, the Central American volcanic arc. All these orogenic systems have built sky island systems, mountainous systems that behave like a sky island. And so I want to talk to you about two specific studies very briefly. We don't have much time, so I won't go into methods details that encompass these temporal and spatial scales I've been talking about. The first one is the PhD research of Sergio Perez Consuegra. He did his PhD in my lab. He works, he's a researcher in Universidad de San Carlos in Guatemala, and he's also the mammal curator. And his study was about the phylogeography and diversification and local adaptation of a mountain rodent group of rodents, the Peromyscus mexicanus species group, that distribute along the Chiapas-Guatemala highlands. And this is the region that we sampled for this study, and he found many incredible things, but I'll mention a few ones related to these complex mountain systems. The first one is that he found very high species richness, diversification, much higher than the 9 species recognized of this rodent group at the time. All of these are different lineages that could eventually be different species. And these species distribute along an elevation gradient going from sea level up to 4,000 meters along all these different mountains. And we can very broadly divide these group of species like the lowland species and the highland species. And this incredible diversification along from the Pleistocene that increased during the Holocene is related to many factors, but one of the interesting ones is the glacial cycles. We all know that there are glacial cycles on the temperate mountains, but these cycles also affected the tropical areas like the Guatemala and Chiapas highlands, where during the last glacial maximum, we had very narrow lowland, very dry tropical habitats and much more extended cold, wet habitats, and species adapted to these different ecosystems. And that during the Holocene, the interglacial, the lowlands expanded in area. They became less dry and tropical habitats less dry, and the more cold, wet ones reduced the area. And species had concert consequences of these changes. And one of those is demographic changes that we actually detected in our study, meaning that in this graph at the bottom right, we have a time scale on the x-axis going from the past to the present, and the vertical line defines the finish of the last glacial maximum. And these demographic processes that I'm saying that will be concordant with these cycles are reflected in the red ones are our lowland species, some of our lowland species, that actually they show an increase in population size that was expected as this tropical lowland habitat expanded during the glacier. And on the contrary, the highland species, they should be decreasing in size because their area of distribution shrunk. So that's exactly what we are seeing here, a decrease in population sizes. So all these dynamics, we can explain them through this sort of analysis. But also we can talk about things more contemporary. these different highlands, of course, are related, have different vegetation types. The tropical forest dominates on the lowlands, while on the highlands we have from cloud forest to pine forest. And these species, this great diversification also reflects this heterogeneity in the habitat. And just to give you two examples, these two species, these two lineages, FG, they are, one is restricted to the lowlands, the other is restricted to the highlands of the same mountain. And they even though they are sister species, they have very different ecological patterns and morphological patterns. So we see this diversification within a mountain. And another example, we can compare also two highland species inhabiting different mountains, but mountains that are very close together. I don't know if you can actually see here, but it's 8.5 kilometers separating these two mountains, these two populations, and they are tremendously diversified. They're different in every sense: size, ecological aspects, what they eat. So all of these myriad of factors are contributing to this diversification. So this will be one of the study. And I want now to tell you again very briefly of another study now in the Trans-Mexican volcanic belt. This is the work of master's thesis of Gabriela Borja Martinez, a student of mine in the Instituto de Ecología. And her study, her research was studying the landscape genomics of quasi-endemic rodent species, the black-eared mouse, quasi-endemic to the Trans-Mexican volcanic belt, where she wanted to decipher how the heterogeneity, the habitat, the environmental heterogeneity governs the dispersal and connectivity patterns of these different populations of these species. And she worked on La Malinche Volcano within the La Malinche, which is a national park that you can see here. And there's many things to highlight here, but I want to mention two. One, that she worked with the remote sensing methodologies and techniques in order to be able to build 2- and 3-dimensional data regarding the landscape variables she studied. And this 3-dimensional, you'll see, becomes to be very important in this study and analysis. And she worked in 3 geographical scales: one global, encompassing two different facing slopes, one north, one northeast, and elevational gradient within each slope going from 2,800 to 3,500 meters height. Firstly, we found that in a global scale, the 2 of the intermediate elevation floors of one of the slopes separated from the rest. So this is the first pattern. But then going more regionally, more locally, within each of the slopes, and using a whole battery of analysis that allowed to see in a more local scale what variables of the landscape are the most important determining differentiation between individuals and populations. We found for one of the north-facing slope that it was tree height, the variable that was determining the separation between the more elevated, the higher elevation individuals and populations from the lower elevation. And tree height was related to a vegetation change along this gradient. And in the other, the northeast slope, it was another variable. It was aspect, which has to do with solar radiation and aspects of the terrain that differentiated the intermediate elevation individuals and populations from the rest, and also allowed to identify a couple of genetic breaks that are linked to a very narrow dirt road, which is not very used or anything, but it has an effect on populations along this slope. And when we come to combine the entire set of data, namely genetic data, environmental data, these 2 and 3D landscape variables, to test hypotheses about different connectivity, we found something really interesting. The global hypothesis was where you see the lower connectivity, you of course have the highest differentiation between or among populations, all to the point to where you can have isolated population along this landscape. And this is exactly what we found. And we also know, or are able to know, what are the main variables combined in combination that are making this differentiation, these changes in connectivity. And for one of the slopes is, again, the variable tree height, but in combination with another, this is an index of vegetation that are separating or that identify how the highest elevation levels are more isolated. Whereas in the other slope, it's again tree height, but litter cover was the combined important variable. And this is very, very interesting because it has to deal with the ecology, how this rodent it feeds and covers for temperature control, and these variables separated or identify more isolated population at intermediate levels. And to conclude with all of this study, it's actually to say that this forest environmental heterogeneity across the different geographic scales determines, based on different variables, landscape and local variables, the dispersal, the genomic structure, and the connectivity patterns in this rodent. And we can translate this in many things about ecology and genetics of species. And the other thing that I want to highlight again is that the fine scale topography that we were able to evaluate with the 3D vegetation structure is a key variable that controls different aspects that we would not be able to model or to identify with 2-dimension data. So this is 3-dimension data is very new in this field, so but it's very, very good. And to conclude, then, I hope I can very quickly convince you that this environmental complexity that drives patterns of ecological and genetic variation are not only key to understand the species diversity and the distribution in both the temperate and tropical sky islands, and that if we really want to truly understand evolutionary and ecological dynamics that at the end lead to speciation and to local adaptation that we can information that we can apply to conservation, we really have to work on getting as much detail and as much information as we can with these many different tools and strategies. And with that, I'll thank you all.
Thank you, Ella. Thanks very much. Okay, so now we'll move with Jessica, and then remember that we'll have some time for questions and answers, so please be prepared. Okay, I introduced Jessica already, but just in case that you want to hear, Jessica Perez Alquicira is a full-time professor at the University of Guadalajara, working on population genetics, phylogeography, and focusing mainly on plants like agave, physalis, guadua, and brachea. Jessica, welcome. Thanks for being here.
Thank you very much. Okay. Well, my talk will be very specific in one species, Oxalis alpina, and the presentation is the influence of climatic transition at the end of the Pleistocene on the distribution of genetic diversity and breeding system of Oxalis alpina in the Sky Island region. I will talk about the results of 2 main papers, and my main collaborators is Cesar Dominguez, Francisco Molina from UNAM, Mexico, Olga Tusko from Kentucky University, Stephen Weller from Irvine, California, and he has been the main person working on Oxalis, and the first work that he published was in 1976. Oxalis alpina is an herbaceous, perennial, and tetraploid species that inhabit in the Sky Island region above 1900 meters in the Sky Island region. This region has been, the distribution of Oxalis alpina in this Sky Island region have been the consequence of the weather warming that during this transition at the end of the Pleistocene. And before that period, during the last glacial maximum, 18 years ago, this region presents higher precipitation and lower temperatures. And according to the fossil records, particularly in the Neotoma-Gudrat Midden, the coniferous forest and Oxalis alpina, because it's associated to these woodlands, presents a lower elevation and lower latitude. So the populations were more connected probably than the current distribution. Therefore, this climatic transition have impacted the evolutionary dynamics of species through the reduction of gene flow and the presence of bottlenecks and further events. Under this historical context, we analyze the impact of climatic oscillation on the distribution of genetic diversity and breeding system of Oxalis alpina. Oxalis alpina is a very interesting, has a very interesting reproductive system. It's a Tristyly species that is characterized by the presence of 3 floral morphs. Each morph is differentiated from the others by the position of the stigmas. This is the female part in relation to the 2 levels of anthers. And we have the long morph, mid morph, and short morph. Short morph has the stigmas below the 2 levels of anthers. And according to the genetics of Tristyly, we expect that each floral morph be in the same frequency. And associated to these polymorphisms in the stigmas and anthers, they have an incompatibility system that doesn't allow the self-fertilization or fertilization between different levels of anthers. Therefore, only the compatible crosses are called legitimate, occur between the anthers and stigmas that are located at the same level. So the white arrows indicate the legitimate crosses. However, we have seen that in the Sky Island region, particularly in this region, Oxalis alpina present modification of the incompatibility system. Long and mid morph are completely cross-compatible. It means that the 2 levels of anthers of the short morph can fertilize ovules from the long morph, and also the 2 levels of anthers can fertilize the ovules from the short morph. So this completely cross-compatibility makes the mid-morph in disadvantage. And we have seen that if the incompatibility differentiation among long and short morphs reduce, the consequence is a reduction of this morph. So natural selection is against this morph when the long and short are completely cross-compatibility. This is the distribution of the morph frequency in the Sky Island region. We have a very marked or strong latitudinal pattern where southern population are tristylos and northern population are distylos, where in the distylos populations, the mid-morph is absent. So under this historical context and the distribution of the breeding system of Oxalis alpina, our main question was to understand the role of natural selection and a stochastic process associated to climatic oscillation on the evolution of breeding system and genetic diversity of Oxalis alpina. We used 2 molecular markers, DNA chloroplast sequencing, in order to see the genetic structure and gene flow via seeds, and nuclear microsatellites using, to analyze the genetic structure and detect gene flow via pollen. So the main result from chloroplast DNA, we detect 8 haplotypes and a very high genetic structure. GST is 0.73. and 5 genetic groups were detected in these populations, and it's clear a latitudinal pattern of genetic structure. No? We can see the red haplotypes at southern ranges, green and yellows in the central part, and blue haplotypes at the northern ranges. And the most northern ranges, we can see this pink haplotype. Associated to this latitudinal gradient of genetic structure, we detect a negative relationship between latitude and genetic diversity. So it means that the northward migrations as a consequence of the warming weather at the end of the Pleistocene have produced reduction through the bottlenecks and founder events in these northern populations. Ecological niche modeling also support these results. We have here the distribution during the last glacial maximum at southern ranges, and the present distribution occurs in the northern ranges. The intersection is the blue color, and we can see that the intersection of past and present distribution is very, very small. But this supports the previous results. But not only the climatic oscillation have impact the distribution of genetic diversity, but also the breeding system of Oxalis alpina, since we can see here in the northern parts of the distribution the loss of mid-morph and therefore the evolution to Tristyly condition, and we see that in this graph that latitude is a negative relationship with the mid-morph frequency. So we conclude that genetic drift associated to founder events may have influenced the reduction of this morph frequency and therefore the evolution to Tristyly condition. Result for microsatellites, we detect two main clusters based on population network approach, and also the structure analysis give us two main clusters. And if we analyze the genetic structure within each cluster, we detect for the cluster 1, 4 different groups, and for the cluster 2, 8 different groups. Unlike chloroplast markers, microsatellites indicates a gene flow among the whole distribution, where, for example, haplotype in brown is in different latitudinal, it doesn't have a pattern, it's an intermixing. So, and the GST was lower than the chloroplast markers, was 0.26. So additionally, we detect using this grouping approach phenograms of Oxalis alpina. In this first phenogram, we have DNA markers from chloroplast, and here are from microsatellites. And we can see that the distylus populations have probably different Tristylus ancestors. So most probably Tristyly evolved within each mountain separately, several times. So our conclusions are that we detect a higher genetic structure with the DNA chloroplast, and therefore gene flow is more restricted to the seed than pollen. The genetic signatures suggest northward range expansion of populations, and similar patterns of these genetic structures can be detected in other species, like tree frogs and jumping spiders in this region. We suggest that natural selection has promoted the reduction of mid-morph frequency, and after that, a stochastic process, particularly genetic drift, associated to these founder events have contributed to the reduction or maybe to loss of mid-morph frequency. The unique historical and evolutionary features of the Oxalis alpina populations in the Sky Island make it an invaluable model system to study evolution on plant reproductive system. And this scenario of the Sky Island allows us to test in hypotheses of the role of fragmentation and isolation of populations in the evolutionary dynamics, not only of the neutral, based on neutral markers, if not adapted traits. And it seems, according to several studies, that adapted traits are the more divergent characteristics that can be found in this sky island. The financial support was provided by CONACYT, NSF, and UC Nexus. So we thank all these institutions. Thank you.
Thank you. Gracias, Jessica. Thanks very much. All right, so we have about 4 minutes for questions. So please type them in the chat box. The first one that I have here is from Barbara. I believe, please correct me if I'm pronouncing it correctly. It says, from what you said, landscape genetics allow us to see which parts of the landscape allow for population connectivity. If you had GPS tracking data, would it not produce similar results, or does it add more information that would not be able to be obtained simply through GPS tracking?
Okay. There will be two differences mainly. One is, despite you may have the GPS, you will have the location of the individual. I don't know what tracking you mean, that you will have the same individual in different trackings, so you will kind of know how it disperses. But what you need to actually relate the landscape variables, the landscape environment and heterogeneity that is influencing how your individuals move and how population structure. You need to have a measure of dispersal. Genetic information has that advantage, even though it is indirect measure of dispersal. It is also the main information where individuals have a potential to adapt, to evolve, even at that scale, even at the individual scale. So combining genetic information with the environmental and doing this evaluation, how one influences the other, that is why you wouldn't be able to do that only with GPS. But you know what's the crown of the queen? When you have the three things, when you have the genetic data, when you have radio tracking, so you have actual movement measures of your individuals that you can also combine with the genetic information that will corroborate or not that dispersal, but will give you the evolutionary information of your study and the environment. So that's that. We've done that in a few rodents. So my answer in short is no, you cannot do exactly the same thing with only GPS data.
Gracias, Barbara. Yeah, I think we might have maybe time for like a 1 quick minute question answer in case there's anyone else. From Robert Arnold, a question from Ella. Fascinating results. Clearly, we don't have remotely sensed data for the future. Nevertheless, I wonder what we can learn about habitat land use for species, which will be useful for in future prognosis in conservation and management. Ella, maybe very quickly.
Yeah, well, yeah, we don't have the remotely sense, but we've actually also tried to do that with species distribution models going into the future. Actually, Sergio, in his last chapter of his thesis, which is not yet published, he modeled these different lineages that we found with all the different characteristics in highlands and lowlands, and how they will be potentially affected not only in their distribution, but in their genetics and their demographics. So that's one way you can do it.
All right. Thanks very much to everyone for this semi-session. I think we're out of time. There's another question from Alicia, but maybe we can type some answer, Jessica, on the chat box so that we can move on. The next session, I will introduce John first, and then I'll wait for Alicia's talk after. John Veens, please correct me again if I'm not pronouncing it correctly, is a professor at the Department of Ecology and Evolutionary Biology at the University of Arizona. He's interested in evolution, ecology, biogeography, and phylogenetics, especially the origins of biodiversity patterns and responses of species to climate change. He strives to address topics that span all or most living organisms, but his favorite study systems are reptiles and amphibians. So welcome, John. Thanks very much for being here. All yours.
Thank you. It's great to be here, and I really appreciate y'all inviting me to participate, and I thank everybody very much for showing up. So I'm going to sort of ask maybe the stupidest question in my talk, which is, why are there sky island distributions? And I'm going to do this focusing on one lizard species in our sky islands that we have here in Southeast Arizona. So when I think of sky islands, I think of this old book by Friedrich Gelbach, Mountain Islands and Desert Seas. So here we are around Tucson, of course, and our sky islands are this set of mountain ranges south of the Colorado Plateau, but north of the Sierra Madre Occidental. That's sort of the ones that I'm thinking of, like this beautiful Cave Creek Canyon here in the Chiricahuas. And for lots of different groups of organisms, these sky islands here in Southeast Arizona have lots of montane specialists. So these are found just in isolated patches of Madrean forest. And this is shown in these green patches below this famous vegetation map. So the big question that I want to focus on is, how do we get to have a pattern like this? How do we get these things that are in these disjunct populations in different mountain ranges? Why are there sky islands? So, sort of, I think the obvious answer I think that most people would give is that this pattern comes about because in the past it was cooler, right? And these Madrean forests and their flora and fauna were distributed continuously across the region. And then the climate warmed up and things were forced by local extinction up into these different mountain ranges. Okay, so that's the basic hypothesis that I want to test today. This goes back to some old stuff that I talked about like 17 years ago, talking about niche conservatism and speciation. And the specific hypothesis that I want to test today is that the montane species that we see today were once widespread in the lowlands when climate was cooler, and then they went locally extinct at lower elevations as the climate warmed. And they did that. They went locally extinct rather than adapting to the changing conditions. Okay, and in some ways, this is sort of maybe a precursor to what's going to happen as the climate warms today. So I wanted to test this idea in this one species of lizard, the Euro-spiny lizard, also called the mountain spiny lizard, Scolopus euroei. And in Arizona, it has this typical Madrean sky island distribution. It's got a much more widespread distribution in the Sierra Madre Occidental, but I'm going to focus on the processes that are just happening from mountain to mountain here in Southeast Arizona. And it's a really great study system because it's active all year and it's easy to find. Bottom line is the thing basks on big rocks when the sun shines. It could be in January, it could be in July. As long as the temperature is more or less okay, it'll be out there. So that makes it really easy to find. And that's in contrast, just jump back here to all these other species, which are much harder to find. So these lizards are really hard to find, which makes them easy to find, excuse me. These Euro-spiny lizards are really easy to find, which makes them a really nice study system. The other thing that's kind of nice about them is that there's this congeneric species, Scolopus clarkii, that occurs at lower elevations, tends to occur at lower elevations in the same microhabitat. So that actually gives us a positive control for the absence of Scolopus euroei. Basically, when you're seeing clarkii, the euroei aren't going to be there. Okay, so what did we do? We did field surveys. So we actually documented in detail where does Scolopus euroei occur across these mountain ranges in Southeast Arizona. So we did elevational transects on basically almost all mountain ranges in Southeast Arizona that have at least some Madrean woodland. We did biogeographic analyses trying to answer whether current climate or possibly competition with Scolopus clarkii might explain the distribution of Scolopus euroei, both elevationally and which mountain ranges it's on versus which mountain ranges it's not on. And then we did population genomics. We tried to make a time-calibrated phylogeny among the populations and try to see if this was consistent with a pattern of local extinctions related to climatic warming. And then finally, we did physiological analyses. We wanted to test whether there was stasis or local adaptation in the thermal traits of these lowest elevation populations across different ranges. And the sort of the basic the basic idea behind this sort of a model of niche conservatism, local extinction, and separation of populations of sky islands is it sort of implies that the physiology is stasis and not is the physiological variables are exhibiting stasis and not exhibiting rapid changes to the changing climatic conditions. If they were, you wouldn't get this pattern. Okay, so that's what we did overall. Just to worth tell you the details here. So we did elevational transects on 31 mountain ranges in southeast Arizona, basically all of them with Madrean woodland. Okay, and we found that Scoloporus euroei was present in 18 mountain ranges, 16 that also had Scoloporus clarkii, and there was another 13 mountain ranges that had Scoloporus clarkii only. To show you what some of these places look like, it's the Patagonia Mountains, very nice Madrean woodland. But there are other places like the Johnny Lion Hills, much smaller mountain ranges with just a few oaks. They only qualify as having Madrean forest because they just have a couple of random oaks there. And this also, the Paria Mountains, looks like a typical desert mountain ranges, but there's a few scattered oaks at the top. Also with Sky Island, and we actually discovered that Scoloporus euroei was there, hadn't been reported before. Okay, so overall, we found that Scoloporus clarkii occurs at lower elevations with warmer, drier climates. We do statistical analyses to show that using this climatic data set from Wang et al. But here, let me show you the interesting thing is that mountain ranges without Scoloporus euroei clarkii actually occurs at higher elevations. So here I'm just showing you all 31 mountain ranges that we surveyed. The black dots show you the maximum elevation of the highest peak in each mountain ranges. The red is the highest elevation that clarkii, in which we found clarkii. The blue is the lowest elevation where we found Scoloporus euroei. And so the interesting thing here is that on those mountain ranges where Scoloporus euroei is absent, Scoloporus clarkii, the low elevation species, goes up to much higher, significantly higher elevations. So what this actually suggests that it's Scoloporus euroei that determines the lower elevational limits of clarkii and not vice versa. We actually did behavioral studies and we can show that Scoloporus clarkii is actually significantly shyer than Scoloporus euroei. Scoloporus euroei is actually bold. In some cases, you can walk up to them and stick your finger right in their faces. Scoloporus clarkii, we documented you basically can't get within 10 or even 20 feet of them sometimes. Okay, so then what explains the absence of Scoloporus euroei on mountain ranges with Madrean forest if it's not Scoloporus clarkii? So we found that it's not competition, it's not current climate. You do ecological niche modeling, all 31 mountain ranges have suitable climate for Scoloporus euroei. What we found is that the highest sites with Scoloporus clarkii and ranges with no euroei actually have similar climate to the lowest sites with Scoloporus euroei. So basically they're similar, the places without euroei are climatically suitable for it. So what we actually found is that the maximum height of the mountain ranges predicts the presence of Scoloporus euroei. Basically, almost if you look here, almost any range that gets above 2,000 meters has Scoloporus euroei. You can just show this with logistic regression. And it's these lower elevation mountain ranges that have just Scoloporus clarkii. So we can sort of make sense of that. And what we think is that these were basically, Scoloporus euroei was pushed off the top of these mountain ranges where the climate was warmer, and the populations went extinct rather than adapting to warmer conditions. Okay, moving right along then, the population genomics. We obtained Scoloporus euroei tail chips from about 1 to 14 individuals per mountain range, mean of about 5 or so, from all 18 mountain ranges in southeast Arizona where it occurs. So we looked at 119 individuals using the RAD-seq genomic approach. We looked at about 1,495 loci and about 4,491 variable sites. And we built an evolutionary tree for the different populations on different mountain ranges using maximum likelihood with RAX-ML. And by using an external calibration from other Scoloporus, we were able to do a penalized likelihood, a time-calibrated tree. Here's just the things to show you. We found that many clades match islands of higher elevation habitats over 1,500 meters. So that corresponds to this light green color. So basically, if you look, you can see these 5 islands of higher elevation populations, and they actually correspond to these 5 clades on the phylogeny. We also found that almost all of the mountain ranges are monophyletic. So all of the individual samples from the mountain range represent a monophyletic group. except for a couple mountain ranges which are not separated by low elevation habitat. And so this is the pattern that you would expect if there was local extinction, killing off the low elevation populations, forcing the montane species, this montane species up into the sky islands and doing it gradually. So they do it in terms of these islands of higher elevations. We also found, you should look at the time scale on the tree here. If you look in the lower right, something really interesting here, these clades diverged over 1 million years ago and not during the Pleistocene glacial cycles. And this has also been found in various other taxa of arthropods and some other groups. So this is all happening. This separation of these sky islands is actually happening over a time scale of more like 1 million, about 1 million to 4 million years, not in the past 20 or 10,000 years or so. Okay, the physiology then. So we looked at physiological variables from Scalopus europaei and Ton mountain ranges in 2016. We focused specifically on the populations near the lowest elevation site in each range. So this could be relatively low, like in the Dos Cabezas, or relatively high up, like in the Huachucas. And this could be sort of, oaks mixed with mesquites, like in the Dos Cabezas, or a mixture of pine and oaks, like in the Huachucas. And so we sampled these mountain ranges that were accessible by road and that span different lower elevation limits. You'll see why we need to do this by road in just a minute. And then we measured body temperatures, basically shoving a thermometer up the lizard's cloaca. And we also did this thing where we looked at voluntary thermal maximum. This is a relatively novel physiological variable where we specifically look at the temperatures that they avoid, which we think is actually much more relevant than the critical thermal maximum. How do we actually do this? Well, you take 4 lizards, you attach a thermometer to their inner thigh, you put them in these metal cans, and then you attach a wire around the metal can, and it slowly heats up. And there's a piece of plastic over the top of the can, and you have the computer monitoring at the temperature at which the lizard pops out of the can. And this is actually highly repeatable and ends up making a lot of sense. Long story short, I'm probably running out of time here. This mean voluntary thermal maximum is mostly invariant among the 10 sites, and it's unrelated to local climate. So we have, for example, here's mean annual temperature, here's a voluntary thermal maximum, very similar across these different sites, across these 10 sites, very different from the Scalopus clarkii, the low elevation species that was recorded in 2 mountain ranges occurring sympatrically. So basically this suggests stasis in this physiological variable rather than local adaptation. And then finally, these body temperatures were actually negatively related to the climate of the habitat, the opposite of the prediction of local adaptation. So as the annual temperature got warmer among the sites, the body temperatures actually got lower. Okay, so in conclusion, we think that these results generally support the idea that the sky island distributions are explained by the extinction of lower elevation populations in response to past climate change. So these biogeographic analyses show us that the height of the mountain ranges explains which mountain ranges have Scalopus europaei and which ones don't. And it's not current climate, it's not competition, and it's consistent with extinction in the lower mountain ranges. We did population genomic analyses, which show that the groupings of elevated ranges are consistent with the extinction of lowland populations over time. This actually occurred a surprisingly long time ago, maybe 1 to 4 million years ago. And then we did physiological analyses that suggested the populations in different mountain ranges do not seem to adapt to different climates at their warm edge, that is the lowest elevation populations. And this is consistent with a failure to adapt to a changing climate over time. The bottom line is that these Madrean sky islands in Southeast Arizona seem to show this footprint of widespread local extinctions caused by global warming in the past, and this is what's created this basic sky island distribution pattern. Okay, so our current research, just mentioned one last thing, is we've been actually resurveying these populations to see if they've been impacted by the recent changes in climate that have been going on. We've looked at about 8 of these 18 populations so far, and it's sort of a mixed bag, but they're actually mostly doing okay, especially in the hottest populations. They actually seem to be holding on to their current range. With that, I'd like to thank all my wonderful co-authors for their work on this project, especially Agus Camacho, who helped me collect the physiological data, Teresa Jeskova for help with the GIS, Elizabeth Miller, Shay Lambert, and Jeff Stryker for helping to collect and analyze the RADSEEK data, my wife, Ramona Walls, who came with me on almost all the field surveys. And if you're interested in reading more about this, this is published in Molecular Ecology a couple of years ago. And with that, I'll be happy to wait for questions.
Thank you, John. Well, so we are a bit behind now for 2 or 3 minutes. So I'm gonna, I guess if it's okay with everyone, we can just, we'll give at least a little bit more time too, and then we'll just try to either eat somewhere else or just push the whole thing. So it is my pleasure now to introduce Dr. Alicia Mastretta Llanes. She's a researcher in Mexico's National Commission for the Knowledge and Use of Biodiversity, CONABIO. Broadly, her research aims to incorporate evolutionary processes into the conservation and management of Mexican biodiversity. Her research covers topics ranging from the effect of topography and climate fluctuations in shaping population genetic structure, to the genetic implications of domestication and human management of crops, crop wild relatives, and trees. I had the pleasure to know Alicia for a while, but actually never met. So we're living in a perpetual state of COVID. But I'm really excited to have her here. Alicia, welcome. All yours.
Thank you very much. Can you please confirm if you are looking at my screen and my presentation? Yeah, yes. Good. So thanks everyone for the invitation and the great opportunity to present here along with other great speakers. The title of my talk is From Genetic to Species Diversity in the Trans-Mexican Volcanic Belt Sky Islands. And in today's talk, I'm going to try to summarize one of my main research goals, which is examining the micro-macro evolutionary link to explain the high endemism of tropical sky islands, focusing on the Trans-Mexican Volcanic Belt. Here in this picture, I'm sitting at almost the mountaintop of La Malinche, which Ella mentioned earlier, and I'm looking at Ixtaccíhuatl and Popocatépetl volcanoes, which Paulo worked with and also mentioned earlier. In this talk, I will try to present the results of 3 main research questions we have been addressing the last couple of days. Years. So first question is if populations of sky islands persist and differentiate in the same mountain through the Pleistocene grassland cycles as a function of topography. That is, if a population can persist in the same mountain during the interglacial and glacial periods, just at different altitudes. And also, can you see my cursor here? Yeah, okay.
Yes, we can.
Also, if depending on the surrounding landscape, populations of different mountains could have been connected during glacial periods or not. So to address this, we divided it into sub-questions. The first is to examine if alpine grasslands persist in a single mountain. We looked at bibliography research on literature research on pollen records, and we found that for these mountains, the alpine grasslands seem to occur at the same mountains during the glacial and interglacial periods, just at different elevations, around 1,000 meters different. We also developed climate niche modeling, and as you can see here in blue, the alpine grasslands persist both in the present and the glacial and interglacial periods. So in summary, based on climatic modeling and fossil pollen records, both favor long-term in situ persistence of populations. To explore what this implies for population and demographic histories, we developed RADS data of these 2 alpine shrubs, Berberis alpina and Juniperus monticola. And then we tested with the allele site frequency spectrum whether they share a demographic history. We basically found that almost all of the populations of both species share demographic events of expansion or stable size, which happen at the same time, which means that these are congruent with long-term population persistence in sky islands. That is, species, the populations of these plants are sitting there in these mountains, going up and down a slope, but not getting extinct within the single mountain. To test how does historic connectivity given by topography influence population genetic differentiation, we developed isolation by resistance analysis using flat distances, but also rasters with the present distribution or interglacial distribution of the species given by niche models or altitude, and similarly, but for glacial distributions. Shortly, we found that for both species, the glacial distributions better explain population differentiation. So in summary, for the first question, populations of sky islands indeed persist and differentiate in the same mountain as a function of topography. The second question we have been willing to address is a little bit more complex. It's basically whether colonization of newly emerged mountains happened from other mountain tops, either by long distance colonization or by glacial connectivity during the glacial stages, followed by allopatric differentiation, or if instead colonization of newly emerged highlands occur from lower lands, followed by parapatric differentiation. To test this, I had a master's student, Adriana Uscanga, who went to all these sky islands and sample beetles and spiders at 2 different altitudes representing alpine grasslands and the forest below them, which then we used to sequence independently each one of the individuals that fell into these traps. So what we were expecting for the first hypothesis will be that otus will be distributed in a single vegetation type, but in different mountains, and that genetic differentiation among individuals of a single otus will occur based on mountains. For the second hypothesis, we will expect otus to be present in both vegetation types, but phylogenetically close otus to be present also in each of these vegetation types. So what we found based on the distribution of otus among the different types of habitat is that otus are mostly found only in one ecosystem or another, but not in both, which favors the first hypothesis. When looking at the phylogenetic trees, we found that otus are formed by individuals from a single vegetation type, but that are distributed among different mountains, which again favors the hypothesis of colonization from other mountains and subsequent differentiation. And finally, if we looked at a single otu and the distribution of genetic diversity and structure within it, we found that genetic differentiation among individuals of this same otu are different based on mountain of origin, which again favors this hypothesis. So we didn't find support for colonization from lower lands followed by parapatric differentiation, but we wanted to test this further with a species that inhabits a single mountain but in an altitudinal gradient. For this is a work of Christian Quintero, another master's student, who tests this hypothesis in Lacaria trichodermophora, this lovely fungus, which we sampled in 3 different sky islands at these 2 elevations, and we developed a GBS data set. With this, we tested for population structure and loci under selection. We found a structure based on mountain, and despite that we tested all by different ways for loci under selection comparing altitudes, we couldn't find evidence of selection regarding altitude. This doesn't mean that there is no selection regarding altitude, it's just that this is not something that is reflected genome-wide. So in summary, we found that higher allopatric than parapatric differentiation is present in the genome of Lacaria trichodermophora. So based on our data, we found that colonization from other mountain tops followed by allopatric differentiation just seems to be more frequent than a parapatric differentiation. Now I'm going to move to the third and last question, which is if this differentiation that we have been looking at caused, sorry, I have to have a sip of water. The third and final question is if this differentiation that we are looking at the genetic level on populations of the same species can accumulate from the genetic level to the species level, and in this way help to explain the high level of endemicity that there is in sky islands. So a bit of background, neutral theory of biodiversity, what would be expected here is that dispersion plays a key role in distribution patterns of species. Also that there is a spatial temporal continuum of biodiversity, which is reflected in a fractal hierarchy where the same processes should be seen at the gene, the genealogy, and species levels and higher lineages. If this were true, it would be expected to find a correlation between differentiation found by neutral genetic markers and distance at different hierarchical levels. This is something that was tested before by Baselga and colleagues at continental distances within Europe. And we decided to, and they found that the same patterns of differentiation that are seen at the haplotype level can accumulate and are seen at higher clustering levels up to species levels and higher. So we wanted to test the same ideas, but within a single sky island. This is the Nevado de Toluca Sky Island, where Nancy Galvez, currently my PhD student, established a lot of pitfall traps, again, in different locations within Nevado de Toluca. And then we developed a DNA metabarcoding data set where we examined from the haplotype to higher clustering levels, lineages, species, and higher of 8 orders of arthropods. What Nancy found is that similarity decays with distance in a fractal manner from the haplotype to higher clustering levels, including the species, which is more or less around 3% clustering level to higher ones. And interestingly, this happens both in Diptera, which are good dispersers, and in Colembola, which are poor dispersers, as well as in other 6 other orders of arthropods. So yes, differentiation caused by topographic variable can accumulate from the genetic to the species level, and we think that this is contributing to explain the high level of endemic species present in sky islands. In summary, to conclude, tropical sky islands foment the long-term in situ persistence of biodiversity within the sky islands themselves. Allopatric differentiation seems to play a more prominent role than parapatric differentiation. And differentiation processes driven by dispersal limitations, which are just neutral processes, can accumulate from the haplotype to the community level. In summary, the sky islands of the Trans-Mexican volcanic belt, for the reasons listed above, are cradles of biodiversity from both the genetic to the species level and higher. Thank you very much. I hope I didn't rush too much. I was a little bit concerned about the time. And I'm very happy to take your questions and looking forward to the discussion.
Thank you very much, Alicia. That was amazing. Okay, so we have time for questions. We're, the whole thing has shifted between 2.5 and 3 minutes, but I think we're good. So yeah, go ahead, John.
Okay, so it's 1 million. So Alicia asked, is 1 million years still within the Pleistocene cycles? Isn't it that the lowland extinctions happen recurrently? Yes, I guess the problem is actually that they seem to have distributed like around 5 million years ago, and then a lot of the splitting happened from this time frame of about 4 to 5 to about 1 million years ago, which basically it doesn't seem like the recent more recent Pleistocene climatic fluctuations had much impact on the patterns of genetic diversity that we see. It seems like the overall time frame is much older.
Thanks, John. There's another one. Oh, sorry, Alicia.
No, I just asked the question before you say 4 to 1, because first say only 1. So I thought 1 million years is still between the Pleistocene. So,
yeah, go ahead, John.
So there's 2 questions about how old the sky islands are, and it's a great question that I neglected to address. And the long and short of it is that they're much older. These mountain ranges are much older than the lizards and what's happening. I think overall it's the time frame is more like 25 to 10 million years, and that what's going on with these lizards is happening from about 5 to million years to about 1 million years. So it seems like the mountains have been sort of more or less stable during this time period.
There's another one there for John I don't know if you can see John, from Alberto Rocha. Fascinating talk. I wonder if the orogeny of Atlantic islands might have influenced the genetic structure results you report with your lizards. Do the divergence post-date information? Okay, I guess this is something that we were talking about.
Yeah. So Robbie asked about the interactions with S. magister that limits Euroea at lowest elevations. So the bottom line is that magister is a desert spiny lizard that's sort of down in the desert zone, and in between the Euroea is like the pine oak zone. In between them is the Clarkii, which would be like in the grassland in the lower oak. So basically the magister and the Euroea never meet. You have kind of Clarkii that's in between them.
Thanks. There's a comment by Jennifer Frey. We previously also found that island age was driving mammal diversity patterns in the Southwest, not area of isolation. And thank you for some, there's a link for that paper up there. Okay. All right. Well, in the interest of time, moving on.
Can you say something quickly about
yes,
mountain age? I think that the Trans-Mexican volcanic belt is particularly interesting because the highest volcanoes that we consider sky islands are all of them younger than 1 million years. So I just wanted to highlight that because I think it's interesting.
Very interesting.
Thank you.
Yeah, it's true. It's a much newer system. Okay, there's John and Alicia. To what extent do the error bars on divergence times overlap both and among lineages, and two with the 1 million year better discussed?
So can I answer quickly on the what we have seen in the I love it because in the Trans-Mexican volcanic belt, for many species that today exist at high elevations, the divergence times are prior to 1 million years old, which is before the mountains even existed. So this means that something in the previous landscape configuration was causing already some differentiation. And then when the volcanoes emerged and were colonized, then, well, evolution continued to occur, but the differentiation based on geography at least, and what we have seen was already kind of like there, but based on previous landscape configurations that we don't know how exactly were they.
Yeah, sorry, just something quick, but isn't it true? So in the case of the transvolcanic belt, yeah, it's 1 million years of the current structure of the volcanoes, more or less, but that most of them are stratovolcanoes, which means that we had high peaks before them, just in a different configuration. No, so they might be.
No, but so we have highlands and stuff happening, but having them as huge as they are and reaching those altitudes to provide this habitats, it's a 1 million year thing, because before they were more like calderas and these other things that Luca Ferrari describes very well.
Yeah.
So yeah, it seems I have asked this question to Luca Ferrari, who is my geologist of confidence, and the massive things we see today, they are 1 million years.
1 million years.
Some are a bit older, but in general, it's 1 million. Yeah.
Cool. Alrighty. Well, so we are moving now into our breakout sessions. You all received the question that we're discussing now. You can see the links here for the corresponding groups. So please join there, and we'll see you all here back in a little bit, about 20 minutes or so. All right, group 2 with John.
Yeah, well, obviously we're not able to address the question of how species diversity was in different sky islands was determined by speciation, extinction, and colonization, or the conservation implications of that. I mean, we talked about some things such as, you know, latitudinal variation in sky islands and things like, or the taxon dependence of different sky islands, but we weren't able to estimate the relative frequencies of the importance of speciation, extinction, and colonization, or the conservation implications of those results.
I guess that's all right. We can always go back to those notes. But if there's anything you think that you might want to add?
Maybe Alberto, do you want to add anything?
No. It's a very spiny question to answer, and so we just had a very nice discussion about dispersal abilities and all that. But as John said, it's extremely difficult to answer that question at the moment because the paucity of the data.
All right, group 3 with Nathan.
Yeah, hi. Yeah, we had myself, Dakota, Aya, Jessica, Nick, and Kimberly in our group. And yeah, I mean, it's a big question, but it was nice for stimulating discussion. We ended up kind of focusing on different timescales at which those 3 processes would take effect. Specifically thinking about, what are the cofactors that would contribute to speciation? So thinking about local adaptation, we had some discussion about that, kind of as perhaps leading to incipient speciation in some cases. We're also talking about vigility, kind of per species differences in traits that would lead to dispersal. And, we had a nice discussion about kind of maybe there's a sweet spot, relative to a species body size, where it can disperse between islands and then sort of get stuck, and that could lead to speciation. And then lastly, we were talking about conservation in the context of really we need to try and maintain corridors to whatever extent is possible, although I guess a lot of these sky islands are already quite isolated. And so it's sort of the challenge is applying the sort of theoretical understandings that we're able to gain from comparing taxa to obviously all the practical concerns of life in the desert and the sky islands. So I think that summarizes it. I don't know, Dakota, do you have anything else you want to add?
No, I think that about covers it. Thanks, Nate.
Cool.
Thank you. And yeah, it feels that as complex as answering this is, maintaining corridors is just a safe bet, regardless of what we can learn. So group 4 with Brooks.
Yeah, thanks. Yeah, some similar topics, I think, to what has already been discussed. A couple of different things I think we talked about. I think we sort of focused on traits and, just coming back to the idea of dispersal ability and that sort of thing, and how that can even having proxies for dispersibility and those sort of core traits can really be difficult for some groups, like we especially talked about invertebrates or insects that might have pretty good dispersal ability, but it's not something that's necessarily immediately apparent through something like body size. And so, just kind of reiterating this issue that, yeah, it's going to be difficult unless we can find proxies, whether that's genetic differentiation, kind of within or across the species or some way to help draw generalizations. And then even beyond that, yeah, maybe because there's sort of all that variability, maybe, focusing at larger scales, looking at latitudinal variations or variation in patterns across the sky islands in some of these processes might be a good sort of place to start without having to kind of wade into the species specific information right from the get-go. So.
Great, thank you, Brooks. All right, group 5 with Erica.
Yeah, so I think my group touched pretty much on very similar things as the paucity of data. I think everybody has to talk about like different metrics as to how we can compare these processes across taxa. One of the things that was mentioned that was a bit different was the need to account for other processes that aren't necessarily ecological evolutionary, so like neutral processes that don't really have any influence, and geological processes, which I thought was really interesting because we don't think, yeah, I see Nick rooting over there. So as biologists, a person in my group said that we don't really think of landscapes as evolving and changing, but they most certainly do, and like also thinking about that. another thing that was brought up that was really important from the conservation aspect is that, you know, considering the temporal mismatch in which these processes take place, and then what we're able to do as conservation practitioners and how policies and conservation strategies are implemented, and what that might imply for actually trying to preserve biodiversity, and if that's even an achievable goal, as pessimistic as that might sound, but, you know, keeping a little bit of a reality check. So I think those are the big highlights from my group. And thank you guys for being awesome and having a really great conversation.
Thank you, Erica. Well, we had on group 6, we were the Spanish-speaking group, and we had Marilyn, Stephanie Mata, Cesar Fagundes, Zaida Areri, and America Aranza. Most of us from Mexico, I think, and Cesar from Uruguay. And you know, interestingly, we only had time to tackle a part of the question, but one of the things we were mentioning is that, yeah, the importance of considering dispersal, you know, as one of the main traits that across different like species, and how that might affect some of the other processes that we consider as important for conservation and research. Now, we were also, I guess we had a little bit of discussion too on the mismatches between or the use of baselines in terms of how, you know, whether the species arrived there before geologic processes started happening, or glacial processes started happening. And we basically were just posing the question of how important is that for really understanding how we see speciation and other processes. So we have actually quite a bit of time, and you know, I don't know. I guess I was wondering throughout these different talks, one of the things that I been thinking a lot about, because we face this every day as we're working, you know, like when we are trying to apply for grants and we were trying to tackle the issue of climate change, which seems really, really difficult for organizations to actually deal with it because of the scale of the problem. But at this point, we feel pretty desperate and frustrated, and there's more and more talk about what in some circles has been considered as restoration, right? And we're basically trying to just get ahead of the curve, and just, for example, I've participated in some experiments with California blue oaks, where we take genotypes from the southern distribution of the oak and start planting them next to northern genotypes under different treatments and see how well they perform. But so, you know, we're behind. This is all late, and I guess I would like to listen to some discussion on like how allowed, you know, how much of a green light conservation practitioners have from you guys, from geneticists and experts on biogeography and phylogeography on just moving things around. And I know this can be like a difficult, challenging topic, but I don't know if that's something that is interesting to all of you. Yeah.
So I'm not a huge fan of moving things around. Actually, I will advocate against it, at least when this is done not even as an experiment or to see if it actually works better in a different locality. I think the problem in Mexico is that things are being moved around without even realizing that this is happening. Yeah. Especially because, as you probably know, CONAFOR doesn't have a track record of where the seeds were produced and where the plants are going to go. So I think it might not be necessarily bad. Which I think it's bad is doing it without even recognizing that this is happening. Sure. So that will be like a red flag that I will put to practitioners, because if we even know, if we had this record of where things are being moved, then we could use them as natural experiments in a way to see if this is actually useful or not.
Yeah. Yeah, I guess, yeah, that's true. I actually never thought about that, Alicia, about how CONAFOR yeah, they don't keep track of any of that. And I actually just requested some plants from them to plant in Querétaro, and I have no idea where they're coming from. But yeah, I guess, you know, my question perhaps is more for like organizations like in the case of Skyline Alliance, or more informed organizations where we're trying to see, and this is at a mountain scale, for example, is it okay if we take, you know, a few Mexican blue oaks from the lower edge of the distribution and maybe shift them up, or from south aspects, you know, and collect the seeds from there and then take them to north facing aspects. That's a smaller scale perhaps, or and keeping track of all that. But I don't know what you think about that kind of stuff.
I like the idea that Aya brought up in our discussion group about trying to integrate landscape genetics, and to a point that also Robbie made, that species are not going to be uniform across their entire geographic range. There's going to be slightly different processes that operate there. But I think, as Aya mentioned in our group, the landscape genetics aspect of that is going to be really crucial for your question, Paulo, about, you know, south-facing aspects versus north-facing aspects. Like, there might be different sort of genetic processes going on there that I know nothing about as a paleontologist, but it would be interesting to sort of, it's good to compare those though.
Okay.
Well, there is this work by the group of Cuauhtemoc Science in Michoacan, in which they have moved upward several species of pine trees, 100 meters above the highest range, and they found that there was a better establishment for those species when moved along with the nurse plants, but not when the nurse were absent. And this is a really interesting feature, showing us that biotic interactions that are rarely studied have to be taken into account, and probably play the major role in allowing species to establish higher in the mountains or in new environments, not necessarily islands. But this study of Cuauhtemoc is really interesting and enticing in the sense of showing us that to assist plants to invade or to use new environments, they need, in the case of pine trees, their associates, in this case the nurse plants, under which they recruit.
Yes, thank you, Alberto. That's actually a really interesting thing that I've seen happening also in the Poponista, with ABS encroaching a little bit into Hawaii pine, but almost always growing very close to them.
The other thing is that if, several years ago in Tenerife, in the Canary Islands, they discovered that the former forests that were up in the Macaronesian archipelago were logged, and the forest disappeared, and it became much more drier. And they made many efforts of restoration with seedlings and small saplings, but they were all unsuccessful until they discovered that they have to put really tall saplings that will be able to catch the fog and irrigate themselves. This is a wonderful example of the biotic interaction with the physical environment, because before they were planting small conifers that were not able to catch the fog and irrigate themselves with the fog. But once you put saplings 2 meters tall, they were able to persist, and is highly related to the Peter Grubb's regeneration niche.
Yeah, that's a great point. And I wonder, I like to hear from you guys also more about the difference between taking these sort of approaches of moving things around between plants and animals.
Aya
Because-
Buscus had her hand up earlier.
Oh, sorry, thank you. I can, yeah, go ahead.
Yeah, there you go. Thank you, Paul. Don't worry. Nick framed it very well, that what we discussed about local adaptations and all the differences that you find in lineages that might be the same species, but they are adapting or adapted locally. And I will just like Alicia, I'm totally against moving anything anywhere. And I know you Paul, put it in a framework of these agencies or groups dedicating to conservation and reintroduction. But genetics plays such a key role, and we know it from textbooks of many years ago. If you move any animal that it's adapted, locally adapted to another environment, even though it might look exactly the same thing, it is not, and you will have not synchronizing, for example, reproduction timing, or so many variables that we would think that temperature is the same here than there, but it's not one variable. It's the entire complexity of the system that you need really to take into account. And there are many stories of unsuccessful transplants of animals or plants that have gone bad or terribly bad or not successful at all. That you really moving plants, they have hundreds of years of generation times, which have many particularities. But think about a rodent that has a 2 years life cycle, and so moving animals or plants has to really take into account probably not all the information there is, but the key information. What is your genetic diversity? How differentiated you are? How locally adapted you are? That would be my 3 key things.
I think I like to think about it the way what physicians say, first do no harm, right? Like you have to think about what are the big risks, and there might be a payoff, but you're taking huge risks that could have really bad. It might be better to do nothing than to take the risk.
Yeah. There's Edmundo Gonzalez. This is a great point, I think. He's asking us about what do you guys think about leaving plots within skylands vacant for a time until regeneration occurs naturally and see what happens. And I think this is a particularly interesting question in the context of fire regimes, which is very different from the tropical skylands down there in central Mexico than here. But that is something that is definitely a very interesting question. Okay, some people have to go. Alicia, I love this question and would like to have separate workshop. Yes, we were just saying, Alicia and I were just, I think this is, honestly, this would be extremely valuable, not only for us, but I think for a lot of practitioners, if we could get together with the expertise that you guys have. And this is especially about mountains, right? I think we agree on that. And if we could actually draw some general conclusions and general maybe laws or some rules about how to do these experiments and how to do these approaches, because it is really challenging for us whenever there are grant applications for climate adaptation. Honestly, it's always a mess, and so we probably should actually get together just for this question at some point. All right, well, we're there, 101. Thank you everybody for joining us again. I really appreciate it. Thanks for your patience and your questions and your answers and everything. So we'll see you one last time next Friday. We'll have a super interesting discussion again, and have a good weekend.